Hi everyone, my name is Lindley and today I'll be discussing DNA mutations. We will begin with the given wild-type DNA sequence. The top strand that reads 5' to 3' is the coding strand, while the bottom strand, which reads 3' to 5', is referred to as the template strand. Following transcription of the DNA strand, you can see the resulting mRNA strand. As translation occurs on the mRNA strand, protein gets created. These proteins are methionine, the start codon, tryptophan, isoleucine, threonine, and the stop codon. The first mutation we will review is missense mutation. Missense mutation is when an amino acid undergoes change to become another amino acid. As seen bolded here in the coding strand, the G in the original sequence stands for the protein tryptophan. Once transversion occurs in the mutant sequence, the G becomes a T, and that T gets changed to a U in the mutant DNA to mRNA sequence. This is identified as a transversion mutation since purine gets changed into a pyrimidine. The translation which the mRNA sequence gets changed into creates the proteins shown here. As you may have noticed, the serine that was created through this mutation type replaced the tryptophan which previously stood in its place. The next mutation will be a nonsense mutation. Nonsense mutation occurs when the amino acid within the sequence gets changed into a stop codon. In this type of mutation, the G in the normal sequence will undergo a transition mutation into an A in the mutant mRNA sequence. As a result of this transition, the proteins created are methionine and the stop codon. Because the stop codon gets coded earlier than in other mutation types, this form of mutation has a tendency to create shorter polypeptide chains. It is also important to mention that this transition mutation occurs due to a purine becoming another purine. The next mutation we will discuss is a silent mutation. A silent mutation occurs when the amino acid remains the same throughout this mutation. In this instance, the T from the normal wild-type DNA becomes an A. This change is considered to be a transition mutation as the pyrimidine turns into another pyrimidine. Here, the proteins that get created from the mRNA sequence are methionine, tryptophan, isoleucine, threonine, and the stop codon. So, in this silent mutation, the isoleucine remains in its original position. The last mutation is a read-through mutation. A read-through mutation happens when a stop codon gets changed into an amino acid. This causes for the chain to lengthen, creating for a longer polypeptide. In this example, T is the U in the UGA stop codon. Through the transition that occurs, the former UGA becomes a C in the amino acid CGA. This codes for the protein arginine and takes the place of the previous stop codon. I hope that you have found this video informative and helpful. Thanks for watching.